Hello dear multicellular organisms, welcome to our class. In today's class, we are going to discuss a topic that's an activity that is a Hydrilla experiment. This experiment is also known as Hydrilla experiment, or else this experiment is also known as to prove oxygen gas is liberated, and it is also known as to prove photosynthesis must require presence of light. So these are the different names of this experiment we are going to discuss. So here requirements: we need boiling test tube, glass beaker, water, funnel, hydrilla twigs or elodia twigs. So both of the twigs we could utilize here, or else anyone, no problem. So procedure: first of all, we have to take the beaker, fill the beaker with water, 3/4 of the beaker. So later, into the funnel, hydrilla or elodia plant twigs should be placed. After placing, we have to invert this funnel into the water which is present in the beaker. Next step, we have to take the boiling test tube, and 3/4 of the boiling test tube is also should be filled with water. Just 1 or 2 centimeters gap must be air space. Okay. So same as the funnel, the test tube also along with water should be inverted on the stem of the funnel carefully. So we have to make sure that or ensure that 1 or 2 centimeters gap or air space should be inside the boiling test tube after inverting on the stem of the funnel. So next we have to place this entire setup in the presence of sunlight, 2 hours or 4 hours, 2 to 4 hours. So such type of setup 2 we have to prepare. This is setup A, and one more setup B also we have to prepare. Setup A we should be placed in the presence of sunlight, and the setup B we have to place it in the dark room. So sunlight should be absent. So after 2 or 4 hours, if we could observe these both setups, the setup in which is placed in the presence of sunlight, in that setup inside the boiling test tube, water bubbles formation clearly it will be visible. But in setup B, which is placed in the dark room, no water bubbles formation. So that is a proof that in the presence of sunlight photosynthesis takes place. After the photosynthesis, at the end of the photosynthesis, oxygen gas is liberated in the form of bubbles. But in setup B, this is absent because sunlight is not provided to that. And how can we conclude that the bubbles are oxygen bubbles? In the sense, carefully we have to take out this test tube, and the water should be eliminated immediately by closing the mouth of the test tube with the help of incensed or lightened matchstick. If we test this gas, slightly the flame of the matchstick or incensed matchstick will be raised within fractions of seconds. So according to the chemical nature and characteristic feature, the gas which is responsible for raising the fire, that is oxygen, and the opposite gas is CO2, that will put off the fire. So with the help of incense matchstick, we can conclude that the produced gas is oxygen. In this way, by doing this experiment, by following the procedure, we could conclude that oxygen gas is liberated in the photosynthesis. So this is the Hydrilla experiment, or also called Hydrilla experiment. So dear multicellular organisms, in one more video we will meet with our one more topic. Thank you.